Welcome back to the deep dive. This is the place where we dig into, well, sometimes entire biochemistry chapters and pull out all the essential knowledge so you can walk away feeling like you really get it.
And today, we are tackling what I think is probably the most critical process in all of cellular biology. We're talking about oxidative phosphorylation.
Right. This is the absolute core of how you turn the food you eat into the energy that actually keeps you alive.
The usable currency of life,
Yeah.
which is ATP-adenosine triphosphate.
And before we even touch the mechanisms, the how, I think we need to talk about the scale of this. The numbers are just, they're staggering.
They really are. It's almost impossible to intuit. So if you're a sedentary adult, just going about your day, your body needs about 83 kilograms of ATP.
83 kilos. That's more than I weigh.
It's more than most people weigh. But here's the catch. If we could somehow pause everything and check, you'd find you only have about 250 grams of ATP in your entire body at any one time.
Okay, so that just doesn't add up. How do you square that circle?
It means that every single ATP molecule in your system is being recycled. And not just once or twice. It's being recycled around 300 times every single day.
300 times. Wow.
And oxidative phosphorylation, or OP as we'll probably call it, is the massive, incredibly efficient engine that makes that recycling happen.
So that's our mission for this deep dive. We're going to trace that whole energy conversion journey. It's the final and really the most powerful step of what we call cellular respiration.
Exactly. It's a beautiful chain of cause and effect. The cause is, oxidizing your fuel, the glucose, the fatty acids. That generates these high-energy electrons, which get stored in NADH and FADH.
And the effect we want, the end goal, is to make all that ATP. But there's this incredible process in between. The electrons, they flow downhill, energetically speaking.
They do. They flow all the way to their final destination, which is molecular oxygen, OR. That whole reaction releases a huge amount of energy. It's very, very exergonic.
But, and this is the key bit, that electron flow doesn't just directly make ATP. That would be too, I don't know, explosive.
Right. It's not a direct chemical link. Instead, the energy from that flow is used to do something mechanical. It powers the pumping of protons across a membrane.
So you're building up a gradient.
You're building an electrochemical gradient, which we call the proton-motive force. That force, that stored energy, is what then physically drives the molecular motor that makes ATP.
So it's this amazing transformation. You go from chemical energy in fuel to electrical energy in the electron flow.
Then to a kind of stored potential energy in that proton gradient.
And then finally back to chemical energy in ATP. That's the roadmap. Let's start with the power station itself.
The mitochondrion.
Right. We all get taught in high school that it's the powerhouse of the cell. But that phrase really hides the fact that its physical structure is everything. The architecture makes the whole process possible.
Oh, absolutely. The structure is the system. Mitochondria are these oval-shaped organelles, about the size of bacteria, maybe 2 micrometers long, and they're defined by their two membranes.
Let's start with the outside one, the outer membrane. It's pretty simple, right? Almost like a sieve.
Yeah, it's quite permeable. It's full of these large protein channels called VDAs, which stands for voltage-dependent anion channels. They basically act like open gates for any small molecules or ions.
So things like phosphate, nucleotides, they can just pass right through from the cytoplasm into that space between the two membranes.
Exactly. But the inner membrane, that's where the real action is. It's the complete opposite. It's a fortress wall. It's incredibly impermeable, especially to charge things like protons.
And it's not smooth. It's all folded up, isn't it?
Extensively folded. These folds are called cristae, and their whole purpose is to increase the available surface area. Massively.
And the numbers on this are wild. The source material estimates that the total surface area of the inner mitochondrial membranes in an adult human is something like 14,000 square meters.
14,000. It's about the size of 3 American football fields. Just think about that. All of that membrane packed inside you is just dedicated to housing the machinery for this one process. It shows you the scale we were talking about.
So because of these two membranes, you get two distinct compartments. There's the inner membrane space, which is between the outer and inner walls.
Right. And then there's the matrix, which is the very center, the space inside the inner membrane. And that's where the citric acid cycle happens, feeding electrons into the system.
And the separation is key, because the whole point is to pump protons out of the matrix and into the intermembrane space.
Precisely. So the intermembrane space becomes positively charged and acidic. We call it the P side for positive. And the matrix becomes negatively charged and more basic, the N side. The inner membrane is the dam holding that potential energy back.
Okay, so that unique structure, the two membranes, the fact that they have their own DNA, This all points to this fascinating origin story, the endosymbiotic hypothesis.
It's one of the best stories in biology, I think. Mitochondria are, they're semi-autonomous. They have their own small circular piece of DNA, just like a bacterium. They have their own ribosomes to make proteins.
Which all suggests they didn't start out as part of our
The
cells.
theory is that billions of years ago, an early, simple eukaryotic cell engulfed a free-living bacterium, one that was really good at aerobic respiration. But instead of digesting it, they formed a partnership.
A symbiotic relationship. The bacterium got a safe home and plenty of fuel.
And the host cell got a ridiculously efficient power supply. It was a huge evolutionary advantage.
We can even see the fingerprints of that original bacterium in modern genetics, can't we?
We can. If you sequence modern bacteria, the genome that looks most like our mitochondrial genome belongs to a species called Rickettsia prowazekii, the bug that causes typhus. It's like a piece of molecular archaeology.
But what's even crazier is the evidence that this didn't just happen randomly all over the place. It seems to have been a single event.
A single event that gave rise to every mitochondrion in every animal, plant, and fungus on Earth today.
How can we be so sure about that?
Well, we look at some really unusual ancient organisms. There's a protozoan called Reclinomonas americana. Its mitochondrial genome is like a living fossil. It's much bigger than ours, with 97 genes.
Okay.
And crucially, those genes include every single protein-coding gene that you find scattered across all the other more specialized mitochondrial genomes we've ever sequenced.
So it's like the master blueprint, the original set.
Exactly. If this had happened multiple times, you'd expect to see different sets of leftover bacterial genes in different lineages. But we don't. We see this common ancestral set preserved in Reclinomonas. It's powerful evidence that we all inherited our power stations from one single ancient merger.
Okay, so we've got the power station set up. Now let's talk about the fuel itself. Just how much energy are we really getting out of these NADH and FADH molecules?
To measure that, we need a concept called reduction potential, which we symbolize as E0 prime or E dollar.
And that's just a way of quantifying how much a substance wants to either gain or lose electrons, right?
That's all it is. It's electron transfer potential. You can think of it like
Yeah.
like a diving board. The higher the diving board, the more potential energy. Electrons start on a high board, a negative potential, and they fall to a very attractive target with a positive potential.
And we have to have a 0 point, a reference.
We do. In biochemistry, the standard reference is the hydrogen gas couple, which we just defined as 0 volts at a biological pH of 7.
So something that's a really good electron donor, like NADH, is going to have a negative reduction potential.
Correct. And a really strong electron acceptor, the ultimate one being oxygen, has a very positive potential. The total energy you can get is just based on the difference between the start and the end.
And there's an equation for that, right? For the thermodynamic drive.
There is. It's a fundamental one. Delta G equals minus NF, delta E. The delta G is the free energy we want to find.
Yeah.
N is the number of electrons being transferred. F is just a constant, the Faraday constant. And delta E is that potential difference we just talked about.
So let's run the numbers for the whole chain, from NADH all the way down to oxygen. NADH, the donor, has a potential of about negative 0.32 volts.
And oxygen, the acceptor, is way down at the bottom with a potential of plus 0.82 volts.
So the total drop, the delta E, is 1.14 volts.
Exactly. And when you plug that 1.14 volts into the equation for 1 mole of NADH, the free energy released it's enormous.
Right.
It's negative 220.1 kilojoules per mole.
Negative 220. That's a huge amount of energy. And just for context, to make 1 mole of ATP, you only need about 30.5 kilojoules.
Right.
So that raises a big question. If you release 220 kilojoules, why don't you just make 7 ATPs? Where does all that extra energy go?
And that is the absolute heart of this whole deep dive. The energy isn't used in one big go. It's carefully captured and stored in the proton-motive force, the delta.
So this is the energy that's stored in that unequal proton distribution we mentioned earlier. And it has 2 parts to it.
It does. Both are created by pumping those protons out of the matrix into the intermembrane space. The first part is the chemical gradient, the difference in pH.
Right, because you're pumping protons out, the matrix becomes more basic, and the intermembrane space becomes more acidic.
And the second part is the charge gradient, or the membrane potential, delta psi. You're pumping positive charges out, so the matrix becomes electrically negative compared to the outside.
And we can quantify that stored energy.
We can. Under normal conditions in the cell, you have a pH difference of about 1.4 units and a membrane potential of about 0.14 volts.
Okay.
And when you combine the energy stored in both of those gradients, it turns out that every single proton that gets pumped out stores about 21.8 kilojoules per mole of free energy.
So that's the conversion. The massive 220 kilojoule energy drop from the electrons is channeled directly into charging up this proton battery.
That's it. It's the most powerful and efficient energy storage system in the biological world.
Okay, so let's move from the physics to the actual machinery. The electron-transport chain, or the respiratory chain. It's made of 4 massive protein complexes, I through 5e.
And they act like sequential transformers. They step down the energy of the electrons in stages, and they use that energy at each step to pump protons.
And these complexes aren't just floating around randomly, are they?
Not usually, no. They tend to cluster together into these huge supercomplexes called respirasomes. This makes the whole process much more efficient because the electrons don't have to travel as far between steps.
So before we get into the big 4 complexes, we should probably introduce the smaller mobile carriers that shuttle the electrons between them.
Absolutely. The first one is a molecule called ubiquinone, or coenzyme Q. We'll just call it Q.
And this one is interesting because it's not a protein, and it lives inside the membrane itself, right?
Exactly. It's a small hydrophobic molecule with a long fatty tail that lets it diffuse freely within the lipid bilayer. It's the ferry that moves electrons between the first couple of complexes.
And Q is the thing that really links the electron flow to the proton movement.
It is. It can accept 1 or 2 electrons. And crucially, when it accepts 2 electrons, it also has to pick up 2 protons from the matrix side to become fully reduced, forming ubiquinol or QH0.
And then when it gets oxidized later on, it releases the electrons and dumps the protons on the other side in the intermembrane space?
Precisely. The second mobile carrier is cytochrome C. This one's very different. It's a small, water-soluble protein that lives in the intermembrane space.
So it's on the outside of the intermembrane.
Yes, and its job is to shuttle electrons from complex 3 to complex 4.
Now, cytochromes have a heme group, like hemoglobin, but they use it differently.
Very differently. The iron in hemoglobin's heme binds oxygen. The iron in cytochrome C's heme is purely for electron transport. It just cycles back and forth between its reduced state, Pho, and its oxidized state, Phu, carrying 1 electron at a time.
And inside the big complexes, there's another type of electron carrier too, right? The iron-sulfur clusters.
Right, these are critical for moving electrons within complexes 3, 2, and 3. They are these little clusters of iron and sulfur atoms held together by cysteine residues in the protein. And just like cytochrome C, they carry 1 electron at a time by flipping the iron between Pho and Pho.
And they are so important that if you can't build them correctly, it causes devastating diseases.
It does. There's a protein called frataxin that helps assemble these clusters. If it's defective, you get a disease called Friedreich ataxia, which is a severe neurodegenerative disorder. It just shows how vital this whole system is.
Okay, let's start the chain. Complex I, the NADH-Q oxidoreductase.
This is the entry point for the electrons from NADH, our most powerful donor. Complex I is a monster. It's the biggest of the complexes, a huge L-shaped structure.
And it takes the 2 high-energy electrons from NADH, passes them through a couple of internal carriers,
An FMN group first, and then a whole series of those iron-sulfur clusters you mentioned.
and finally delivers them to our mobile carrier, Q.
But the real magic is how it couples that electron transfer to pumping protons. It's a fantastic piece of engineering.
So how does it work? It's not a simple channel.
No, it's more like a piston. When Q accepts the electrons and becomes negatively charged, that charge is transmitted electrostatically to some key amino acids in the arm of the L that's embedded in the membrane.
And that zap of negative charge causes a physical change.
It does. It causes the protein helices in that arm to shift their shape. It's a conformational change, like a molecular plunger, and that movement physically pushes 4 protons out of the matrix and into the intermembrane space for every NADH that gets oxidized.
4 protons, okay. So what about the other electron donor, FADH0? It takes a different path.
It does. It enters at complex 2, the Succinate-Q reductase. And what's unique about complex 2 is that it's actually also an enzyme from the citric acid cycle. It's succinate dehydrogenase.
So it directly connects that metabolic pathway to the electron transport chain.
Exactly. It oxidizes succinate to fumarate, and the electrons are captured in its FADHers group. From there, they pass through some iron-sulfur centers and go directly to the Q pool, making QHers.
But there's a huge difference here that explains why FADHers gives less ATP.
And that is the critical point. Complex 2 does not pump any protons.
So the electrons from FADHers completely bypass the first proton pump.
They do. They're entering the chain further down the energy hill, so you just get less energy out of them. It's a less powerful entry point, but it's essential.
All right, so now we have this big pool of reduced QHers from both complex I and II. This is the fuel for complex 3, the Q-cytochrome C oxidoreductase.
Right, and this is our second proton pump. Its job is to take the electrons from the 2-electron carrier, QHers, and hand them off to the 1-electron carrier, cytochrome C.
Which sounds like a logistical problem. How do you hand off 2 electrons to a carrier that can only take 1 at a time?
You solve it with a really clever, but admittedly complicated mechanism called the Q cycle.
Okay, this is the part of the textbook that always makes my head spin. Can we just try to get the concept down? What is the point of the Q cycle?
The point is twofold. First, it makes sure you don't waste one of the electrons from QH heroes. And second, it basically doubles the number of protons you can pump. It's a recycling system.
A recycling system? How?
Okay, so 2 QH heroes molecules are used, one after the other. For the first QH heroes, 1 electron goes to cytochrome C, as you expect. The second electron takes a different path and is temporarily stored on another Q molecule that's already in the complex.
It recycles it.
It recycles it. Then the second QH heroes comes in. Again, 1 electron goes to another cytochrome C. And its second electron is used to fully reduce that stored recycled Q molecule, turning it back into QH.
And to do that, it has to grab 2 protons from the matrix.
Exactly. So the net result is that you've passed 2 electrons to 2 cytochrome sane molecules. But in the process, you've pumped a total of 4 protons across the membrane. It's an incredibly efficient way to maximize the proton gradient.
Okay, that makes sense. A bit complex, but elegant. So now we're at the very end. Complex 4. Cytochrome C oxidase.
The terminus. This is where 4 of those reduced cytochrome C molecules come in, one by one, and deliver their 4 electrons to the final acceptor, molecular oxygen. Oro?
And that's what makes the water. This is the whole reason we breathe oxygen.
This is it. And this final step releases a tremendous amount of energy, but it also faces a huge safety problem. You have to reduce oxygen without letting any dangerous intermediates escape.
Because partially reduced oxygen is incredibly reactive and damaging.
Extremely. So the active site of complex 4 is this little molecular cradle made of a special heme group and a copper ion that holds onto the oxygen molecule very tightly.
So it doesn't let go until the job is done, a safe reduction.
Exactly. It waits until all 4 electrons have arrived, passing through a couple of intermediate stages before it finally releases 2 harmless molecules of water.
And of course, it's also a proton pump.
It is. So to make the 2 water molecules, it consumes 4 protons from the matrix. We call those the chemical protons.
Right, they're part of the final product.
But the energy release in this step is so massive that the complex uses the extra power to pump an additional 4 protons across the membrane, the pumped protons. So in total, complex 4 removes 8 protons from the matrix for every 1 Oyurov molecule. It's a powerhouse.
The need for that safe reduction mechanism really highlights the central paradox of life, doesn't it? That oxygen, the thing we need to live, is also incredibly dangerous.
It is, because the system isn't 100% perfect. Maybe 1 to 3% of the time, an electron leaks out and only partially reduces an oxygen molecule.
And that's when you get reactive oxygen species, or ROS.
Right. If 1 electron gets transferred, you get the very destructive superoxide radical. If 2 get transferred, you get hydrogen peroxide.
And these things just wreak havoc. They damage DNA, proteins, lipids. It's the damage that's linked to aging and a lot of diseases.
So the cell needs a really robust defense system. The first line of defense is an enzyme called superoxide dismutase, or SOD.
And its job is just to find that superoxide radical and neutralize it.
It converts it very quickly into hydrogen peroxide and oxygen, which is better, but hydrogen peroxide is still not great.
So that's where the second enzyme comes in, catalase.
Right. Catalase takes that hydrogen peroxide and breaks it down into harmless water and oxygen. SOD and catalase are the cell's cleanup crew, constantly mopping up the inevitable oxidative spills.
But here's the paradox you mentioned. It turns out ROS aren't just purely bad.
Not at all. We now know that low, controlled levels of ROS are actually used by the cell as signaling molecules for all sorts of things, from cell growth to the immune response.
It's that classic idea of the dose makes the poison.
Absolutely. Think about exercise. When you exercise hard, you ramp up your metabolism and you actually produce more ROS. But your body responds to that stress by making more of the protective enzymes, like SOD. So in the long run, the stress makes your defense system stronger.
Okay, so We've used the electrons to charge the proton battery. We have this massive proton-motive force. Now for the payoff, how does that gradient actually make ATP?
This brings us to Peter Mitchell's brilliant chemiosmotic hypothesis, which won him the Nobel Prize. He proposed that the electron transport and the ATP synthesis are two separate events, linked only by this electrochemical gradient.
Which was a revolutionary idea at the time. People thought there must be some direct chemical link.
But he was right. And the proof was so elegant, researchers created these tiny artificial vesicles like little soap bubbles, and they put just two proteins in the membrane.
What were they?
The mitochondrial ATP synthase, the machine we're interested in, and a protein from bacteria called bacteriorhodopsin, which is a proton pump that's activated by light.
So they created an artificial system.
A very simple one. And when they shone a light on these vesicles, the bacteriorhodopsin started pumping protons in, creating a gradient. And instantly, the ATP synthase started cranking out ATP.
Even though there was no electron transport chain at all.
None. That experiment proved definitively that the proton gradient alone is the direct power source for making ATP.
And the machine that does it is ATP synthase, also known as complex V. It looks just like a motor.
It really does. It has a ball part called the F unit that sticks into the matrix, and a stick part, the F unit, that's embedded in the membrane.
The F unit is the catalytic part, right? It's made of this hexamer of alpha and beta subunits.
Correct. The beta subunits are where the ATP is actually synthesized, and sticking up through the middle of them is this central stalk, the gamma subunit, that acts like a drive shaft or an axle.
And the F unit, the part in the membrane, is the proton channel. It has a stationary part, the A subunit, and this ring of C subunits that can actually rotate.
And we now know that these syntheses don't just work alone. They team up. They form dimers and long oligomers, which helps us stabilize them against the incredible rotational forces. And it also helps to bend the membrane and create the sharp curves of the cristae.
So how does this motor actually work? The mechanism is called the binding change mechanism.
Right. And the key insight here, the thing that changed everything, was the discovery that it doesn't actually take much energy to make ATP from ADP and phosphate on the enzyme surface.
So the reaction itself is almost free.
When the substrates are tightly bound, yes. The really hard part, the energy-requiring step, is getting the newly made ATP to let go of the enzyme.
So the job of the proton gradient isn't to force the reaction to happen.
No. Its job is to provide the energy to physically rotate that central gamma stalk. And that rotation forces the catalytic subunits to change their shape, which in turn forces the ATP to be released.
And since there are 3 of those catalytic beta subunits, they cycle through 3 different shapes or states.
Exactly. As the gamma stalk turns, it pushes them through the cycle. There's the L or loose state, which just binds the ADP and phosphate.
And then it rotates.
And it becomes the T or tight state.
Right.
This is where the magic happens. It clamps down, and the ADP and phosphate spontaneously become ATP.
And then one more rotation.
And it becomes the O or open state, which has a very low affinity for ATP. So the newly made molecule just pops off into the matrix. One full 360-degree rotation synthesizes and releases 3 ATP molecules.
And this idea of a rotating motor isn't just a theory, it was actually seen.
It was, and one of the most beautiful experiments in biology. Researchers stuck the EVA part to a slide and attached a long fluorescent actin filament to the top of that rotating gamma subunit.
Like a tiny propeller.
Exactly like a propeller. Yeah. And when they fed the motor ATP and ran it in reverse, they could watch under a microscope as the filament spun around in distinct 120-degree steps.
One step for each ATP molecule hydrolyzed. That's incredible. It's direct visual proof of rotational catalysis.
So the final question is, how does the flow of protons actually turn that wheel?
Right. What's the connection? This happens down in the F part in the membrane.
It does. That stationary A subunit has 2 proton half channels. One opens to the high proton side, the intermembrane space, and the other opens to the low proton side, the matrix.
And they connect to the rotating C ring.
They do. Each subunit in that C ring has a crucial acidic amino acid, usually a glutamate, right in the middle of the membrane. Normally, it's negatively charged and doesn't want to be in the oily membrane.
But to get it to move, you have to neutralize that charge.
You have to protonate it. So a proton comes in from the high concentration channel, binds to the glutamate, and neutralizes it. Now that subunit is happy to rotate into the membrane.
It's like a turnstile. You have to pay a proton to get through.
It's exactly like a turnstile. The ring clicks forward one spot, and it keeps rotating until that protonated subunit reaches the other half channel. the one facing the matrix where the proton concentration is low.
And there, the proton pops off and goes into the matrix, and the subunit is charged again.
And that keeps the rotation going in one direction. That physical rotation to the C-ring is what spins the gamma stalk and drives ATP synthesis up in the FeO unit.
So the number of protons it costs to make an ATP depends on how many subunits are in that C-ring.
It does. One full turn always makes 3 ATP. So if you have, say, a 12 subunit ring, it costs 12 protons to make 3 ATP, or 4 protons per ATP.
But vertebrates are more efficient.
Much more. Our Th rings typically have only 8 subunits. So for us, it costs 8 protons to make 3 ATP, which is about 2.7 protons per ATP.
For our purposes, we can just round that and say it costs about 3 protons flowing through the synthase to make 1 molecule of ATP.
That's a good working number, yes.
Okay, so we've established the intermembrane is this impermeable fortress, which is great for holding a gradient, but it creates a huge logistical problem. How do you get fuel in and products out?
You need specialized transport systems, and there are 2 big challenges. First, what do you do with the NADH that's made out in the cytoplasm during glycolysis?
Right. NADH can't just cross the membrane to get to complex I.
So the cell uses electron shuttles, and there are 2 main ones. The first is called the glycerol-3-phosphate shuttle. It's mostly used in muscle and the brain. It's fast, but it's a bit wasteful.
How does it work?
Cytoplasmic NADH passes its electrons to a small molecule, which then ferries them to an enzyme on the outer face of the inner membrane. That enzyme then passes the electrons not to NADH, but to FAD, making FADH.
Ah, so the electrons then get passed to the Q pool.
Exactly, which means they completely bypass complex I. They miss the first proton pump. So using this shuttle, you only get about 1.5 ATP for that cytoplasmic NADH. You pay a price for speed.
But there's a more efficient way.
There is. In the heart and liver, they use the malate-aspartate shuttle. It's more complicated, involving a bunch of different carriers.
But what's the end result?
The end result is that it manages to use the electrons from cytoplasmic NADH to create a molecule of mitochondrial NADH inside the matrix.
So those electrons get to enter at complex I after all.
They do. And so you get the full energy yield, about 2.5 ATP per molecule. The tissue just chooses the shuttle it needs, speed for muscle, or maximum efficiency for the liver.
Okay, that's getting the electrons in. What about the other problem, getting the ATP out and the ADP back in?
That's handled by one of the most abundant proteins in the entire membrane, the ATP-ADP translocase, or ANT.
It's an antiporter. It swaps one for the other.
It swaps one ADP in for one ATP out. But it's not a simple one-for-one trade in terms of energy. Look at their charges. ADP has a -3 charge. ATP has a -4 charge.
Oh, right. ATP is more negative.
And remember, the matrix is already negative. So it takes energy to push the more negative ATP out into the cytoplasm.
And that energy has to come from somewhere.
It comes from the proton-motive force, specifically from the electrical part, the membrane potential. It's estimated that this transport process consumes about 1/4 of all the energy stored in the gradient. It's the cost of exporting your power.
A necessary tax on the system.
A necessary tax.
Yeah.
And the system is so beautifully organized. The ANT and the phosphate carrier, they actually cluster right next to the ATP synthase, forming a little supercomplex called the APP synthesome to make the whole process as fast as possible.
So after all of that, the complexes, the shuttles, the transporters, what's the final score? How much ATP do we get from one molecule of glucose?
The modern best estimates, once you account for all these costs, is about 30 ATP molecules per glucose.
And out of those 30, only 4 are made in glycolysis and the citric acid cycle.
Right. The other 26, the vast majority, come directly from oxidative phosphorylation. It's the big payoff.
Given how powerful the system is, how does the cell make sure it doesn't just run out of control, burning fuel for no reason? How is it regulated?
The regulation is actually incredibly simple and elegant. It's a concept called respiratory control. And the bottom line is that electrons will only flow down the chain if ATP is actively being synthesized.
So the two processes are tightly coupled.
Tightly. And the master controller, the throttle on the whole system, is the concentration of ADP.
The substrate for the synthase. So if you're resting, your ATP levels are high and your ADP is low.
And if there's no ADP, the ATP synthase has nothing to do, so it stops. If it stops, it's not using up the proton gradient.
So the gradient builds up to its maximum, like a dam filling to the brim.
Exactly. And that creates a huge back pressure. The proton pumps, complexes psi 3 and 4, can't pump against that massive force, so the entire electron transport chain just grinds to a halt.
And that backs up even further to the citric acid cycle.
Right. If the chain stops, it can't recycle NADH back to NAD. Without NAD, the citric acid cycle stops. The entire energy output of the cell is perfectly matched, second by second, to the demand signaled by the amount of available ADP.
But there is one big exception to that tight coupling, a situation where you want to uncouple the system.
Yes, and that is for generating heat, a process called non-shivering thermogenesis.
This is what things like hibernating animals or newborn babies use to stay warm. It happens in a special kind of fat tissue.
Brown adipose tissue, or BAT, the mitochondria in brown fat are packed with a special protein called uncoupling protein 1, or UCP1.
And UCP1 is basically a leak, a controlled short circuit.
That's a perfect way to put it. It's a channel that allows protons to flow back into the matrix without going through ATP synthase.
So you still burn fuel, you still run the electron transport chain, you still pump protons, but when they flow back,
The energy isn't captured as ATP. It's just released as heat. It turns the mitochondrion into a tiny furnace. It's a biological heating pad activated by fatty acids when you get cold.
because this whole system is so central to life, it's also a major target for poisons.
A very vulnerable target.
Yeah.
And you can classify the poisons based on what part of the system they attack.
Okay, so first you have things that just block the electron flow itself.
Right? Poisons like rotenone block complex 1, antimycin A blocks complex 3. And the really famous ones, cyanide, azide, carbon monoxide, they all block the final step at complex 4. The chain stops, the gradient collapses, and that's it.
Then you have things that attack the ATP synthase directly.
Like the antibiotic oligomycin, it physically clogs the proton channel in the F0 unit. So ATP synthesis stops, and because of respiratory control, the electron flow stops right behind it.
And then there's the really nasty category, the chemical uncouplers.
The most infamous one is 2,4,4-dinitrophenol, or DNP. It's a small lipid-soluble molecule that basically does the same job as UCP1. It picks up protons on one side of the membrane and drops them off on the other.
It's a synthetic proton shuttle. It blows a giant hole in the dam.
It does. It completely dissipates the proton gradient. So the electron transport chain runs at full, uncontrolled speed, burning fuel and oxygen like crazy, but zero ATP gets made. All the energy is just dumped as heat.
This was actually marketed as a diet drug for a while, wasn't it?
Tragically, yes, back in the 1930s. And it led to horrible deaths because people would literally cook from the inside out from uncontrolled hyperthermia.
And finally, you can also block the transporters, like the ANT.
Right. Toxins like atraculoside block the ATP-ADP translocase. If you can't get ADP into the matrix, the synthase starves and the whole system shuts down.
So clearly, when this system goes wrong, the consequences are severe. This brings us to mitochondrial diseases.
Which are really complex because the mutations can be in your nuclear DNA or in the separate mitochondrial DNA, which you only inherited from your mother. And the organs that get hit the hardest are always the ones with the highest energy demand.
But the brain, the muscles, the heart, the eyes.
Exactly. A common set of defects are in complex eyes, and they can lead to things like Leber hereditary optic neuropathy, which causes sudden blindness. It just shows how dependent those tissues are on a constant, massive supply of ATP.
But beyond just making energy, mitochondria have another darker role. They're central to programmed cell death, apoptosis.
They hold the self-destruct button. When a cell is too damaged to be repaired, or it's infected, it gets a signal to essentially commit suicide in a clean, orderly way.
And the mitochondria kick off that process?
They do. The outer membrane becomes permeable. It gets holes in it. And that allows proteins that are normally kept safely in the intermembrane space to leak out into the cytoplasm.
And the most important one that leaks out is cytochrome C.
The very same molecule that's a crucial electron carrier. But once it's out in the cytoplasm, it gets a new job. It becomes a death signal.
What does it do?
It binds to another protein, forming a huge wheel-like structure called the apoptosis. And the apoptosis' job is to activate a whole cascade of enzymes called caspases.
And caspases are like a demolition crew?
A molecular demolition crew. They go through the cell and systematically chop up all the essential proteins and DNA, dismantling it from the inside out. It's incredible that the same molecule that's a linchpin of life is also the trigger for death.
So we start this deep dive with this incredible challenge. How do you recycle 83 kilograms of ATP every day? And we followed this amazing path.
From the electrons in NADH and FADH2 through the transport chain to the proton gradient, and finally through the rotation of that incredible molecular motor. All of it, the whole massive system, is regulated by the simple availability of ADP. The stability of it all is what's truly mind-boggling. You think about that little protein, cytochrome C, it's doing the exact same job with the same structure in us as it does in a stalk of wheat. It has been perfectly conserved for over 1.5 billion years.
When evolution finds a perfect solution, it sticks with it.
It does. But I think the most elegant takeaway for me is that this whole unbelievably complex power grid that runs your entire body, all this chemistry and electricity and mechanics, is ultimately powered by the simplest physical principle imaginable.
Which is?
It's just the ability of a thin, closed lipid membrane to store potential energy by keeping a bunch of protons on one side.
A simple concentration gradient. That's all it is at its heart.
That's all it is.
It really makes you wonder, doesn't it, if life could figure out how to harness something so fundamental with near-perfect efficiency, what other basic principles of physics might be out there just hiding massive, untapped potential for solving our own future challenges.
We hope this exploration has given you a fresh appreciation for the constant, quiet, and profound recycling revolution happening inside every single one of your cells.
Until next time, keep digging and keep learning.